Hello, everyone, and welcome back to chapter 9, lecture 4. Let's go ahead and begin. In today's lecture, we're going to be covering neuronal activity. So essentially, this is starting by talking about seeing a shape. Neurons at each level of the visual system have distinctly different characteristics and functions. During neuronal stimulation, each action potential is represented by a strike or a spike that we're able to see over here. In image A, in a 1 second period at this neuron's baseline firing rate, 12 spikes were recorded. In image B, a firing rate greater than the baseline signals excitation. And in image C, a firing rate less than the baseline signals inhibition. Then there are these retinal ganglion cells that we talked a little bit about in the previous lecture. These respond only to the presence or absence of light, not to shape. They have a concentric circular arrangement where there's a center and a surround or a periphery. There are the on-center cells, which are excited when light falls on the center portion of the receptive field. They are inhibited when light falls on the periphery of the receptive field. Light across the whole receptive field produces weak excitation. For the off-center cells, they are excited when light falls on the surround area of the receptive field, and they are inhibited when light falls on the center of the receptive field. Light across the whole receptive field produces weak inhibition. So here's a nice little figure that helps to show that a little bit more clearly. This shows how on-center and off-center ganglion cells tell a brain something about shape. So in figure A on the left side, in the receptive field of the RGC with an on-center and off-surround, a spot of light that is shining on the center excites the neuron, which we can see here as the light begins to strike it. But a light in the periphery or the surround inhibits it. When the light is turned off, the firing rate increases briefly in offset response. A light shining in both the center and the periphery would produce a weak increase in firing. Meanwhile, in figure B, the receptive field of an RGC with an off-center and on-surround light in the center, that actually produces inhibition that we're able to see here. Meanwhile, the light in the surround or the periphery, that produces excitation, and the light across the entire field produces weak inhibition. So then there's these overlapping receptive fields where neighboring retinal ganglion cells receive their inputs from an overlapping set of photoreceptors. The receptive fields of the retinal ganglion cells, they overlap extensively, like we're able to see here, so that any two adjacent fields look at almost the same part of the world with just these small differences. And a small spot of light shining on the retina is likely to produce activity in both on-center and off-center ganglion cells. It's just going to vary on which ones that might be. And that's important because of this luminance contrast, where the amount of light reflected by an object relative to its surroundings. And this allows input from the retinal ganglion cells to tell the brain about shape in some ways, right? Because if we have all of these on-center ganglion cells, we're able to see in this hypothetical population whose receptive fields are distributed across this light-dark edge, that the activity of cells along the edge is most affected relative to those away from the edge that are over here. So information that's transmitted from retinal ganglion cells to the visual areas of the brain does not give an equal weight to all visual field regions, and instead it emphasizes those regions containing differences in luminance. So these are the areas that are along the edge. In essence, These retinal ganglion cells, they send signals about edges, and the edges are used to form the shapes. Now, processing shape in the primary visual cortex, or V1. So the V1 neurons, they receive input from multiple retinal ganglion cells, and the V1 neuron, they also have a much larger receptive field, than the RGCs. These cells behave like orientation detectors. They're excited by bars of light oriented in particular directions. There are also these simple cells in the receptive field with a rectangular on-off type arrangement. So simple cells, they respond to a bar of light in a particular orientation, such as A, where they respond to a horizontal direction, or B, where it's more of this oblique orientation. And the bar's position in the visual field is important because the cell either responds, where it would be turning on, such as here, or it does not respond, and it would be off to the light in the adjacent visual field regions. The simple cells are not the only kind of orientation detector in the primary visual cortex. There are several functionally distinct types of neurons that also populate the region of the primary visual cortex, or V1. There are these complex cells. These are maximally excited by bars of light moving in a particular direction through the receptive field. And there are hypercomplex cells. These are like the complex cells that are maximally responsive to moving bars, and they have a strong inhibitory area at one end of the receptive field. So we've got a couple of figures here that are helping to demonstrate this a little bit more. Here we have the example of the complex cell. So unlike the simple cell's on-off response type pattern, in the complex cell in V1, it shows the same response throughout its circular receptive field, responding best to light bars that are moving at a particular angle. Their response is reduced or absent with the light bar at other orientations. So we can see strong response here when the stimulus is at 45 degrees. See a strong response here as well when it's at 45 degrees and it's continuing along this direction. Now, when we put the stimulus at 60 degrees, we see a weak response. And when the stimulus is at 15 degrees, there's no response. So there is that preference, I would say, for a particular direction of movement. Then in the hypercomplex cells receptive fields in V1, these respond to a moving bar of light in a particular orientation, for example, horizontal, anywhere in the excitatory part of its receptive field. However, the response is inhibited if most of the bar extends into the inhibitory area. And so we can see that here where there is this inhibitory area and there's a baseline response. We introduce the stimulus and there's a strong response. As the stimulus moves along, it continues to have a strong response until most of it ends up being in that inhibitory area where we get a weak response. And once all of it is within that area, there is no response. A little bit more about the receptivity of the primary visual cortex. So a V1 cell responds to a row of ganglion cells in a particular orientation on the retina. The light bar strongly activates a row of ganglion cells, each connected through the LGN to a V1 neuron. The activity of this V1 neuron is most affected by a bar of light at a 45-degree angle. And we can, again, see that here where, so each of these circles is representing one of the receptive fields of ganglion cells. And there's this stimulation of the set of the on-center ganglion cells that's exciting the primary visual cortex neurons through those connections. And we see the strong response there. The light goes through, and where the light's not going through in the same way, we're getting the weaker response from that. Now, the neural circuit in a column in the visual cortex. Now, we have this nice three-dimensional view over here, and the sensory inputs enters the cortical column at the layer of V1 at the bottom, and it terminate on the stellate cells in the layer IV that synapse with pyramidal cells in layers 3 and 5. And the information flow is vertical. The axons of the pyramidal cells leave the columns to join the other columns or structures. So the connection between light striking the retina in a certain pattern and the activation of a simple cell in the primary visual cortex. One responds to a bar of light in a particular orientation, and we can also diagram the retinal receptive fields of complex or hypercomplex V1 neurons using the same type of logic. So a characteristic of the cortical structure is that the neurons are organized into functional columns, and the connectivity pattern in a column is vertical. So the inputs arrive in the layer IV, and then they connect with the cells in the other layers. And this is showing us some of those functional columns that I was just talking about. And we can see here that the adjacent columns, they house neurons that are responsive to slightly different line orientations, forming an array of 180 degrees as it's getting slowly rotated over and across. And that continues down to each of those different layers that we were just discussing. So neurons within the same column respond to lines oriented in the same direction. Each of those columns, right? All the same direction. Whereas adjacent columns house cells that are responsive to different line orientations, and then neurons within the same column respond to lines oriented in the same direction. The adjacent columns house cells that are responsive to these just slightly different line orientations. In the cortical columns of input from each eye, this is called ocular dominance columns. V1 has orientation columns housing neurons of similar sensitivity and ocular dominance columns that contain input from one eye or the other. And we can see some of that here where there's the ocular dominance columns that receive input from the right eye or the left eye. These cells, they are maximally excited by complex visual stimuli, like hands or faces or whatnot. They may be selective to particular faces seen head on, to faces viewed in public, to the posture of the head, and even to specific facial expressions. For stimulus equivalence, that's part of the recognizing that an object is the same across different viewing orientations. Tanaka had found that rather complex features are necessary to activate most neurons in area TE. This include a combination of characteristics such as orientation, size, color, and texture. Neurons with similar but not identical responsiveness to particular features tend to then cluster in some of these columns. And we can see that here. So there's in the temporal lobe, the neurons that are forming the columns that respond to these categories of shapes. The preferred stimuli of neurons in the primary visual cortex are fixed, typically through genetics. And the neurons in the temporal lobe can alter their preference with experience. But the difference between the primary visual cortex versus the temporal lobe. The object is represented not by the activity of a single neuron, but rather by the activity of many neurons with slightly varying stimulus specificities. These neurons are grouped in a column. This finding is important because it helps to explain stimulus equivalence, or recognizing an object as remaining the same despite being viewed from different orientations. With seeing color, there's this subtractive color mixing that helps us to obtain the entire range of colors in the visual world that many of us experience by mixing only three primary colors, the red, the blues, and the yellows. This is a similar property of the cones of the retina um, that were taking place. With additive color mixing, that increases light to make colors. So the lighter the color, the more light it contains. So why would a white surface reflect the entire visible spectrum? Well, that's because when all visible wavelengths are reflected, we end up seeing white. Scientists suspect that color vision also gave great apes a competitive evolutionary advantage in making some of these distinctions possibly. Some of the theories about color, one of the primary theories is this trichromatic theory. And this theory is the explanation of the color vision based on the coding of these three primary colors, the red, the green, and the blue. The color we see is then determined by the relative responses of the different cone types, and this could explain different types of color blindness. Now, there is a limitation here in that there are really these four primary colors that includes yellow, which obviously we don't seem to have evidence that we have yellow cones per se, and it also cannot reflect or explain some of the after images that can exist as some of the cones tire with red and green and blue and yellow and whatnot. Now, while most people's retinas contain three types of cones for that trichromatic vision, this image that we have here illustrates approximation of what people with protanopia center or deuteranopia right see compared to what trichromats on the left see. And you can see just this clear distinction for many of us who are not colorblind, at least, between these images. Now, it's important to note that individuals with either of these forms of color blindness, they still see plenty of color, but that color is just significantly different from what the trichromat's color experience is. And many domestic animals like dogs, cats, and horses, they all have this deuteranopia, which gives them an advantage in seeing objects that would appear camouflaged to trichromats. Military forces often use humans with that deuteranopia to help see through camouflage as well, which is just kind of interesting. It also makes me interested too, because I don't see a huge difference between these two. I don't know, maybe others do. There's just small variations between these two for me. And there's another theory about color, which is the opponent process theory. This is the explanation of color vision that emphasizes the importance of the opposition of colors. So red versus green, blue versus yellow. The opponent processing occurs in the retinal ganglion cells, where there's that on-off dynamic and the center surround receptive fields that we talked about earlier in this lecture. And that's true for about 60% of retinal ganglion cells. And here's a little demonstration of some of those opposing color pairs. So for this exercise, look first at this rectangle on the left. Stare at the center here, that black circle, for about 30 seconds. And you know what, I'll go ahead, I'll start up a timer. I'll let you know when to begin, when to stop, in case you're following along. So get yourself looking at that center circle and begin. Try to keep your eyes focused on that circle as best you can. Don't move your eyes around to get the best results. About halfway there. 15 more seconds. All right, now look at the circle in the white box to the right. Focus on that black circle in the middle, and you likely see this after image of green on the left side and yellow on the right side. Now, why does this take place? That kind of phenomenon. Well, thought is that there's the center of the receptive field that is excitatory in some cells and inhibitory in others. So the stimulation to the periphery has the opposite effect, where the center is responsive to one wavelength and the surround is responsive to another. A red-green sensitive retinal ganglion cell responds weakly to white light on its center and surrounds because red and green cones absorb white light to similar extents, so their inputs cancel out. The cell responds strongly to the spot of red light in its center and the red's paired wavelength green in the surround. It is strongly inhibited by a small spot of green in its center, and the RGC, it responds very strongly to the simultaneous illumination of the center with red and the surround with green. It is completely inhibited by the simultaneous illumination of the center with green and the surround with red. And so with the opponent process, the explanation of color vision, that's emphasizing the importance of these opposing color pairs that essentially is taking place within these cells. These are the neurons they're talking about in the cortical, the cortical region V4. They do not respond to particular wavelengths, but they're responsive to various perceived color. The receptive field center is excited by a specific color, and the surround is then inhibited. This may be important for something like color constancy, where perceived color is constant relative to other colors, regardless of changes in illumination. Then going to some of the neuronal activity in the dorsal stream. The posterior parietal cortex is involved in processing visual information for action. We talked about this before. That's the how-to stream, where neurons in this area are silent to visual stimulation when a person is under anesthesia, interestingly enough. Some cells in this area, they process the visual appearance of an object to be grasped, and these cells, they will fire even when a monkey watches another monkey pick up an object, which again, is interesting to see. So what color is the original? The dress here, you may remember this was real popular a few years back. The dress in the center of the photo is perceived as either white and gold or blue and black, depending on the viewer. So the image has been color balanced. The dress is white and gold on the right, and the dress itself is white and gold, but on the right it appears to be blue and black. And so what color really is the original that's taking place here? And what's interesting is that these unconscious assumptions that end up taking place about the source of illumination of the dress influences the color perception that people have. So people believe that the dress to be illuminated with an artificial incandescent light. They saw the dress as blue, whereas people who thought the dress was photographed in daylight saw the dress as gold. Our assumptions are necessary for the human visual system's color constancy. And again, I'll say personally, as I'm looking at this center original image, to me, I'm seeing the gold and white. I'm not seeing the blue and black. And it's not unless I focus hard on this image that the blue and black begins to appear. And I really have to not look at some of the others, because as soon as I look back to the far left, immediately turns gold and white. So that is lecture 9.4. talk to you soon for the last couple sections.